Welcome back. So, while previously we talked about biochemistry and how atoms come together and how they really make up everything there is on Earth, now we're going to start talking about really what are the larger molecules that are made from these atoms. So, atoms are the smallest thing, but then there's also things that atoms make up that are still very, very small. So, we call these things macromolecules. So, you have the smaller molecules, you have the atoms that are going to make up the elements, and then atoms are going to come together and make molecules. And when we start talking about larger molecules coming together, we talk about macromolecules. And then when you start to talk about macromolecules and we're talking about life on Earth or living things, because this is biology, we're going to talk about organic molecules. And organic molecules, as much as you hear about this word organic, it really means grown naturally when you hear organic in layman's terms or in the everyday world. When we talk about it in chemistry, it means that it involves carbon along with hydrogen and oxygen. So carbon is really what is responsible for making life possible. Carbon is a very important element for living things, for biology. And carbon, along with hydrogen and oxygen, makes up these molecules that are organic molecules. And there are four main types of organic molecules. You have carbohydrates, which, when we talk about them in layman's terms, a lot of times are you know, in everyday discussions you hear about, you're thinking about bread or pastas. Those are carbs or carbohydrates. But really, sugars, anything that is a sugar is going to be a carbohydrate. And then you have lipids, which are fats. So lipids are fats, just another word for fats. You have proteins, and then you have nucleic acids, and these are our macromolecules. So carbohydrates include molecules that function as fuel. So our sugars, which are going to be in milk, candy, breads, fruit, pastas, all of these things have carbohydrates in them. They have some kind of sugar that we're going to be able to break down and use in order to have energy. So they form structures of cells in all organisms, and they are made up of carbon, hydrogen, and oxygen. So carbohydrates are composed of monosaccharides, and monosaccharides are simple sugars or single sugars, you can think of them as. And then these monosaccharides are going to come together and they are going to form disaccharides and polysaccharides, long chains of sugar. So, these single sugars or simple sugars are our monosaccharides, and they give you examples of glucose is a simple sugar or a monosaccharide, fructose, galactose. These are all simple sugars. And then the simplest carbohydrates, including glucose, are these monosaccharides. And as the chemical bonds of carbohydrates are broken down and other more stable bonds are formed, a great deal of energy is going to be released. So by breaking those bonds, we're releasing energy. And that's how we're able to use these sugars as a form of energy. When we talk about seeing glucose in the body, blood sugar has one of three fates. Either it's going to be fuel for cellular activity because we are made up of cells, and the reason we need energy is because the cells actually need energy. Or you can have glycogen is going to be stored, or fat is going to be stored. So you can either use it, store it as glycogen, store it as fat. And we see in the diagram you have glycogen, which is our simple sugars. We create these long chains of these simple sugars, and we're able to ultimately store them in these long chains that can later on be broken down. And when we break them down, we can use those simple sugars and break those bonds in those simple sugars in order to get energy for the cells. So because a pound of glycogen has about 4 pounds of water molecules bound to it, dieters lose a lot of water weight during the first days of their diet. So as you're starting your diet, you're going to shed a lot of water weight to begin with. So glucose in the bloodstream can be used for those three reasons. And remember that glucose is a simple sugar or a monosaccharide, which can come together and form these polysaccharides, these long chains of simple sugars. And carbohydrates are polymers composed of monosaccharides, or they are long chains. That's what they mean by polymers. They are a collection of monosaccharides. And you have examples, typically we talk about polysaccharides, which are these long chains, but you also have just two that are put together, which is going to be a disaccharide. And some examples are lactose and sucrose. Lactose is what's found in milk. That's a disaccharide that we're going to break down into two sugars. You see things like table sugar is sucrose, and sucrose is broken down into simple sugars, and we can use that for energy. Um, the carbohydrates are going to serve as structural material in animals and plants. You have things like chitin, which we don't have in humans, but that is a long chain of a long chain polysaccharide, which is going to make a hard material for things like arthropods. It creates their hard shell for protection. And then you have cellulose as well, which creates dietary fiber for humans, so we don't break it down very well, so it helps us with our digestive tract and really cleans us out, and that's why they recommend having dietary fiber like cellulose, so that it can reduce your risk of colon cancer, helps clean you out and keep you healthy. Lipids are another macromolecule, a molecule that is going to be made up of many different atoms, and it is an organic molecule. It contains a significantly more carbon to hydrogen bonds than carbohydrates, so it's more stored energy within there. They're actually insoluble in water, so water is not going to break them up. They're nice for forming things like waxes, cholesterol, sterols, steroids. You have hydrophobic versus hydrophilic molecules involved when we're talking about a phospholipid bilayer. When we move on and we start to talk about the cell and the cell membrane has this is made up of this layer called the phospholipid bilayer, and that creates this hydrophobic versus hydrophilic molecules where it's able to both attract water molecules and push away molecules. That's what hydrophobic, all right, and hydrophilic means. Hydrophobic, you're going to try to push it away. Hydrophilic, you're going to try to take them in. So fats are made up of glycerol along with fatty acid. So you have this glycerol head and these fatty acid tails. So you have one glycerol, and then you're going to have these three fatty acids that are attached to it, and that's what a fat is made up of. So you have triglycerides, which are fats and oils. So fat molecules contain much more stored energy than carbohydrate molecules, and it's going to store a large amount of energy. A strong taste preference for fats over other energy sources has evolved in animals. So since it is beneficial for us, we tend to like fat. Well, nowadays, fat is plentiful, or calories are plentiful for humans, so we don't really have to worry about getting enough. But once upon a time, when there wasn't enough food, it was beneficial for an organism, for an animal, for a human to crave fats because they wanted to be on the hunt for fats in order for them to get enough calories to survive day to day. There's degrees of saturation. You hear about saturated fats versus unsaturated fats, where each carbon in the hydrocarbon chain is bound to two hydrogen molecules in saturated fats. But in unsaturated fats, you see at least one double bond links two carbon atoms in the hydrocarbon chain, causing the fatty acid to have a crooked shape to it. So molecularly, saturated fats, unsaturated fats, they're both fats, but molecularly they look different, and they are different. You have hydrogenation, which occurs where it's the artificial addition of hydrogen atoms to an unsaturated fat in order to make the fat more saturated. And hydrogenation can improve a food's taste, texture, and shelf life, but it also makes a food less healthful. So you don't want hydrogenated oils. You don't want the foods that have hydrogenation, which is going to create the trans fats. You don't want those things. Cholesterol, phospholipids, they're used to build sex hormones and cellular membranes. Okay, cholesterol, important component of cell membranes in animals. It is good to have cholesterol. As much as we talk about how bad cholesterol is, it is a good thing, but too much of it is a bad thing, and it clogs your arteries, and it can lead to heart attack, stroke, Steroid hormones are important in reproduction, and you see that it regulates sexual development, maturation, sex cell production. Estrogen is going to influence memory and mood. Testosterone stimulates muscle growth. So these hormones have a big effect on what's going on in from one individual to the next. Um, with the phospholipids, they are going to create the cell membrane or the plasma membrane, which is the outside of a cell, and they differ from fats in that they have only two fatty acid chains and a phosphorus atom in the glycerol. So they still have that glycerol head to it, but they only have two tails to it, two fatty acid tails, and then they also are going to have the phosphorus atom attached to it instead of that third fatty acid tail. So you have uh, normal fats, and then you have the phospholipids, or normal lipids and the phospholipids. Proteins are body-building macromolecules, which are important in structure, protection, regulation, contraction, as well as transportation. And then in proteins, they are actually made up of these building blocks called amino acids. So the building blocks of proteins are amino acids, and there's 20 different types of amino acids. So they are what make up the proteins, and the different arrangement of the amino acids is what makes one protein different from another. And in proteins, they still have carbon, hydrogen, and oxygen, which we're going to see in all four types of the of our organic molecules, but it also has nitrogen in it as well. So you have your carbon, your hydrogen, your oxygen, and your nitrogen in your amino acids and in your protein. So a carboxyl group and amino group in all amino acids, that's what's found in all of them. They differ from each other by this side chain that they have. Okay, so all of them are going to have this amino group. They're all going to have this carboxyl group right here. They're going to have a carbon attached to a hydrogen, and then they're also going to have this side chain, and the side chains can differ, and you see, all right, if the side chain is like this, it's going to be alanine. If it's like this, it's glycine. If it's like this, it's tryptophan. So all of these all the changes that happen in just this arm of the amino acid is going to change what type of amino acid it is, and then the different amino acids that are going to come together to form a protein is going to determine which specific protein it is. So while there's different types of amino acids, there's also different types of proteins. So the different types of amino acids that come together and the sequence of those amino acids determines what the protein really is. And proteins are essential for growth, repair, replacement, as well as energy storage and bonds. Each different protein has a different composition of amino acids. Like I said, some amino acids must be obtained from our diet, which are considered essential amino acids. Other amino acids we're able to make on our own. And when we start putting the amino acids together to form proteins, they actually start to come together in a certain way, and it makes them have a certain shape to them. So when we start putting them together, we call it our primary structure, how we're piecing the different ones together. And then that ultimately is going to form a certain shape, which is going to be our secondary structure. And then from there, we have a more complex shape, which is going to be our tertiary structure. And then finally, those tertiary structures are going to come together and create a even more complex shape, which we call our quaternary structure. With proteins have a certain function to them, but they are able to actually be broken down. You have environmental extremes such as temperature and pH. All right, proteins only function well under certain temperatures and pH. So once you disrupt that temperature or that pH that it likes to live in, or I shouldn't say live, but that it functions in, it is going to change the shape. And once you change the shape, it's going to change the function of that protein. So we often talk about how things are going to be denatured. All right, there's different types of proteins. Some proteins are hormones, some proteins are enzymes, and enzymes are helpful with completing some chemical reactions in the body. So if you think about something like breaking down lactose, well, you need the enzyme called lactase. Now, most people have lactase and they're able to break down the lactose when they drink milk. Some people don't have that enzyme, they're not able to break down the lactose. And if you were ever to have a situation where the pH or the temperature of lactase, that enzyme, is changed, it's going to change the shape of that protein, of that enzyme, and it's no longer going to be able to function properly. It's no longer going to be able to help with breaking down lactose. So temperature and pH can denature, change the shape of a protein, and when you change the shape of a protein, you're changing its function. Um, Enzymes increase the likelihood for a reaction to occur. So once you ruin that enzyme and you change the shape, it's not able to help increase that likelihood anymore. And enzymes work by decreasing the activation energy. So chemical reactions occurring in organisms can either release energy or they can consume energy. But either way, the reaction needs a little push in order for this initiation to for in order for it to initiate the reaction, and that's called our activation energy. So it needs to get to a certain point and a certain amount of energy for the reaction to take place. And enzymes actually lower the activation energy and make it easier for that reaction to take place. So while it still needs a little bit of energy involved, it needs less with the enzyme there. So the enzyme helps push the reaction along. We call it a catalyst. It catalyzes or pushes along that chemical reaction to occur. So enzymes regulate reactions in several ways, and you're going to see that physical factors can affect and chemical factors can affect the reaction rate of enzyme catalyst reactions. You have the concentration of the enzyme in relation to the substrate, temperature, like I said, pH, as well as the presence of inhibitors or activators that are going to interfere or play a role in the reaction. So here we see just a simple diagram of talking about how much energy you need in order for it for the reaction to take place. So the rate at which an enzyme catalyzes a reaction is influenced by several chemical and physical factors. The rate of a reaction typically increases with the addition of either more enzyme or more substrate. When such an addition causes a higher rate of collisions between enzyme and substrate molecules. So temperature reaction rates generally increase at higher temperatures, but only at the optimal temperature of an enzyme. So enzymes are going to work in a certain area, but once you go past that area, and there is an optimal temperature that they work at, but once you get past that point, it's going to denature it. It will no longer work. So while this enzyme, this is just a hypothetical enzyme, it works between this temperature and this temperature, it works best at this temperature, and once you go past this temperature, it is denatured, it'll no longer work. pH is the same way. While it works best at this, it also can work within this range, but if you go too acidic, it's not going to work. If you go too basic, it is going to denature it and it's not going to work. So these enzymes like to function in specific pHs and specific temperatures. And then you have the presence of inhibitors or activators. All right, reaction rates are going to increase in the presence of activators, and they're going to decrease in the presence of inhibitors. They're inhibiting things or they're activating things. Either they're going to push that reaction along or they're going to hinder the reaction from occurring. Misspelled proteins or incorrect amino acid sequence, putting the amino acid together, it's going to lead to active site disruption, loss of function. So when you put the wrong amino acids together, the wrong protein is going to form, and that protein isn't going to function correctly because the shape is what influences the function. So non-functional enzymes are responsible for many diseases and physiological problems. So you have phenylketonuria, which is one, as well as lactose intolerance. Or if you make the wrong type of lactase, you have a lactase that is not formed properly, then it's not going to be able to break down that lactose, and that is lactose intolerant. And then lastly, we talked about carbohydrates as one organic molecule. You talk about lipids as another type of organic molecules. You talk about proteins as another type of organic molecules. And the fourth one is nucleic acids. And there are two main nucleic acids that we talk about, or the two that there are RNA and DNA. So nucleotides are the building blocks of these nucleic acids. And each nucleotide is composed of three molecules. So you have these nucleotides, and they're made up of a molecule of sugar, which is either going to be ribose or deoxyribose. There's a phosphate group always. You see this here. And then there's also going to be a nitrogenous base. And our nitrogenous bases are going to be adenine, thymine, guanine, and cytosine. And a lot of times you see people just write them as the first letter. So it's going to be A, T, C, G. And in DNA, we know that A is always going to pair up with T, and T is always going to pair up with A, and C is going to pair up with G, and G is going to pair up with C. So nucleic acids carry genetic information. So as much as we talk about um, uh, carbohydrates, lipids, proteins as really making up us as humans and being kind of the building blocks of what makes us us, as well as being a form of energy, nucleic acids are really the directions on how to make us up. So you have DNA, which we'll talk about first, and then you also have RNA. These are the two types of nucleic acids. Both play central roles in directing the production of proteins, but typically we talk more about DNA because that is what it is in our nucleus. So the information in the molecule of DNA is determined by the sequence of bases, where it's the sequence of nitrogenous bases, where it can either be along the sequence, you can have one of four things, and they're all jumbled up differently, and that's what makes the code. But the four things are A, T, C, and G. So DNA holds the genetic information. We're going to talk about this more as we go on, but it's important to understand that you have carbohydrates, you have lipids, you have proteins, and then you also have nucleic acids, which are going to be our DNA and our RNA. So for something like this, if I know that A always goes with T and T always goes with A and C goes with G and G goes with C, and you just give me one strand of DNA, because if you look, DNA is kind of like this twisted ladder. So if we untwist it, we can look at one side and know what the other side is because we know what they pair up with. So we know A always goes with T, C goes with G, A goes with T, G goes with C, and T goes with A. So now if I give you this and I say, well, what is the other side of this DNA? You say, well, C goes with G, C goes with G, C goes with G, C goes with G, T goes with A, T goes with A goes with T, G goes with C, G goes with C, A goes with T, A goes with T, C goes with G, C goes with G. So you knew what the other side of that ladder was just by knowing what one side of the ladder was. And RNA is a universal translator, all right, reading DNA and directing protein production, which we'll talk more about as we move forward. But just understand that there are two different types of nucleic acids. You have your RNA and you have your DNA. So keep these things in mind as we move forward and we start to talk more about DNA and how the molecules really make organisms what they are, how they make up the cells, and how the cells make up more complex organisms.